Well, this is a beautiful evening here on Hood Canal. We had a little snow this morning, but I think it's kind of all gone away, which is really nice. My name is Teri King, and I'm the Marine Water Quality Specialist for Washington Sea Grant that covers Hood Canal and all of South Sound. And tonight's seminar is the 7th in a series of lectures highlighting research being conducted by University of Washington that impacts the economy, lifestyle of Puget Sound. Tonight's presentation is being sponsored by Washington Sea Grant, and I'm pleased to note that our director, Penny Dalton, is here with us tonight, so she wants to raise her hand. I think she was in the back. Oh, she's outside. She's waving her hand back there. Janice McNeil is also in the back. Some of you talked to her earlier today. She was at the office helping out, and she's a great help with us. Tonight we have Dr. Glenn VanBlaricom. He's Assistant Unit Leader for Wildlife at the Washington Cooperative Fish and Wildlife Research Center at the University of Washington. His research focuses on population and community ecology and conservation biology, primarily in coastal marine ecosystems of the North Pacific Rim and Arctic Alaska, which kind of seemed like this morning here. Micah Horowitz, our other speaker tonight, is a doctoral candidate at the University of Washington's Department of Biology, studying under Dr. Jennifer Rusink. Micah's field work has focused on the local effects of geoduck aquaculture on a meadow of intertidal eelgrass in Samish Bay. So, without further ado, we'll start with Dr. Glenn VanBlaricom and the geoduck aquaculture investigations in Puget Sound, digging deep for answers.
Thank you all for coming. It's a great pleasure to be invited, and I want to thank Teri and Penny and all of their colleagues at Washington Sea Grant for organizing the event and making it possible for us to talk about geoducks. What we're going to do tonight is actually 3 presentations sort of squished together into one. I will begin by talking about the study that many of you are familiar with that is looking at the effects of harvest disturbance on the ecology of the intertidal zone in which geoduck aquaculture occurs. Micah Horowitz, as Teri mentioned, will talk a little bit later about his doctoral research on the effects of geoduck harvesting on eelgrass beds in Samish Bay. And in addition, I will be offering a few slides that were sent to us by Drs. Jeff Cornwell and Roger Newell from the Horn Point Environmental Laboratory at the University of Maryland. Some of you may be aware that Roger and Jeff and their colleagues have been working in collaboration with us, sort of side by side in the same study sites, to look at some of the issues relating to the geochemistry of the ecosystem in which geoduck aquaculture is pursued. So they do have some interesting preliminary results, and we'll talk about those as well. So that's the general plan. We have a lot to cover, so I'm going to charge ahead. And Teri, I forgot to ask you, but I assume that we have time for discussion afterwards. Is that okay? I see nodding of heads, so that's a good thing, because I'm sure everyone will have questions and comments. So let's proceed. So to begin with, we'll be talking about the study that I'm directly involved with that has to do with investigating ecological disturbance and ecosystem response patterns in association with geoduck aquaculture operations in the intertidal zone in Puget Sound. My part of the presentation is organized as follows. I'll be talking a little bit about just the basics of the harvest cycle, because it's my understanding that there may be at least some people in the audience that are not, after all, intimately familiar with all the details of the business. So we'll start off with a quick overview of the major phases of the aquaculture cycle. Then we'll talk a little bit about our conceptual context for our study. That is some discussion about natural ecological disturbances and how that type of issue fits in with what we're doing in association with aquaculture here in the Sound. We'll summarize the major goals and aims of our research and our methods. We will review our progress in terms of how we're doing with things like sample processing, quality assurance, the spending of money, which of course we do as fast as we can, and so on. And then finally, we'll offer the first public presentation of some preliminary data, some results from our studies. So that's the general plan. So let's talk first about the business of geoduck aquaculture. Before we do that, this is a list of all the people that are a part of our research team and photographs of some of them in various states of maniacal disarray, which is sort of comes with the territory. It's a fantastic group of people. A few of them are here tonight. and I hope that you'll have a chance to meet and talk with them after the presentations. So to summarize just briefly, the aquaculture cycle begins with two major events: the installation of some sort of predator exclusion device in the habitat in which the culture cycle is to be initiated. And with that comes the outplanting of young geoducks that come primarily from hatcheries. And one of the common patterns is, as shown here, where PVC tubes are inserted into the sediment, and then the young animals are placed within the tubes. And then the tubes are covered either individually with a sort of a net cap, or as we see in this figure, covered sort of collectively with a larger net. Now, I understand that there are a number of variations on this theme, different tube sizes, different densities of tubes, different types of net covering, and so on. And I just want to sort of describe the basic concept here without going into too much detail. This is just one example of the type of predator exclusion apparatus that is typically set up at the beginning of the culture cycle. This is necessary because young geoducks are vulnerable to various kinds of predators, things like crabs. Crabs are a major concern. There may be some losses to shorebirds and a few other predators as well. But it's viewed as an essential part of the cycle if it is to be economically viable to enhance the survival rate of young animals during the first year that they're out of the hatchery and out in the intertidal environment. Let's see, I got ahead of myself here. So the structures typically are left in for about a year. Again, I think there's some variation on that depending on a number of factors, but typically after about a year, the structures are removed. At this point, the geoducks are viewed as being large enough and well enough established that the protection from the predators is no longer needed, so it's removed. So what we're already setting up here is a series of disturbances, okay? And I don't use disturbance in a value-laden way. It's simply a description of a process, and we'll talk more about that later. But disturbance in an ecological context is not necessarily a bad thing. It, in fact, is a very common thing, and I'm using the word in that context. So we recognize several disturbances in the first part of this cycle: the placement of the tubes, and then a period of a year when the tubes are present, which itself constitutes a form of disturbance, then the removal of the tubes after, or whatever the predator exclusion structures may be, after about a year. Thereafter, there is a grow-out period of on the order of 5, 6, 7 years, something like that, depending on growing conditions. It's pretty clear to me that exact timing of harvest is influenced to a large degree by market conditions as well. So there are a number of factors that go into the decision to end the culture cycle by harvesting the clams. But in any case, in the order of 5 to 7 years after the initial outplanting, we have the final disturbance in the cycle, which is the harvesting of clams, typically done, here again we sort of have theme and variations. The removal of clams typically requires that the sediment be suspended with some sort of a water jet so that the clams can be extracted efficiently. I rather suspect that most of you at one point in your lives have tried to dig a geoduck with a shovel, and I can't imagine anything less efficient and utterly hopeless in an economic context. The suspension of the sediment is an essential part of the process, otherwise the entire process would be hopelessly inefficient. So as you can see, a skilled harvester can pull out a lot of geoducks in a hurry. It's actually quite impressive to watch these folks work. So that is the end of the cycle. It's important to recognize from an ecological context that the disturbances that we're talking about are occurring on a number of different scales. For example, there's the smallest scale, which is sort of the disturbance that is associated with the extraction of an individual clam. But there are also larger scale events that have to do with water runoff, placement of large structure fields, and that sort of thing. So there are a number of different spatial scales that are involved as well that tend to make things a little bit complicated, but also very interesting from a research perspective. And then, of course, the product, which is bundled up and shipped off to faraway places, and if everything works well, with a healthy return on the investment, and a little bit of profit and a lot of reinvestment into more outplanting of geoducks. Okay, so with that as background, let's talk a little bit more now about how an ecologist views disturbance. Again, I reiterate that we don't attach value to this process. It's broadly recognized as one of the most significant types of process that can occur in natural communities. Disturbance is defined here by, in Stephen Pickett's classic book that was published about 25 years ago, describing a disturbance as any relatively discrete event in time that disrupts ecosystem, community, or population structure, changes resources, substrate availability, or the physical environment. Okay, so it's a fairly broad definition, and in fact, it encompasses a lot of things. It's a little bit of a catch-all term. There are a number of metrics that we use to characterize disturbance so that we can try to codify the study of disturbance and make it possible to generalize about how ecosystems accommodate disturbance as part of their structure and their process. We're interested in things like how frequent does a disturbance occur, how long does it last. For example, does a fire just burn for a day or does it burn for a month? That sort of thing, because it makes a big difference in terms of ecological response. How big is the event in space? By what chemical and physical mechanisms does the disruption occur? Is it heat? Is it an avalanche? Is it water movement in the form of a tsunami or something like that? So there are a number of chemical and physical attributes that are important to recognize. Probably the part that's most interesting to most of us in our project is the community response after a disturbance occurs. What happens then? What happens after the volcano has blown its top, or the tsunami has swept the beach, or the landslide has occurred? That's the initiation point. What we're really interested in then is how does the system respond? Does it recover? If so, how long does it recover? And what are the pathways of recovery that are taken before we get back to a configuration similar to what was there before the disturbance occurred? Now, the big, rare disturbances tend to attract a lot of attention. We get hurricanes, volcanoes, tsunamis, big fires, and if you're really unlucky, we get a meteor or comet impact. Doesn't seem to happen too often, and I think that's probably a good thing for most of us. These tend to attract a lot of attention, but they are rare events, and in many systems, probably not that important. What is probably much more important are smaller scale events that, in fact, are monotonously common in many ecosystems. I've shown 3 examples here that all happen to include marine mammals because that's something I'm interested in from a research context. We have a dugong in the tropics of Australia rooting around in a seagrass bed, tearing up the bottom, looking for food. A walrus basically doing the same thing in the Arctic. Walruses are capable of blowing water out and then drawing it back into their mouths at very high velocity, in fact causing resuspension of sediments that is eerily similar to what happens at the end of a geoduck culture cycle, except the geoduck guys don't do it with their mouths as far as I can tell. And then the third image there is a side scan sonar output of an area that has recently been worked over by gray whales in the Bering Sea. Gray whales are also bottom foragers. They forage on patches of amphipods and create substantial disturbance on a number of different scales in the bottom where they feed. Most of those individual patches are on the order of a few meters in diameter. So you have the individual patches there and then a large area where there are lots of patches. So there are a couple of different scales of disturbance happening there. But there are a lot of other disturbances, many of which we're probably more familiar with. Tree falls, forest fires, avalanches, mudslides. They all occur with sufficient frequency that they can have very important ecological impacts. And they're part of the system. The key issue then is for anthropogenic disturbances is to try to figure out the extent to which the disturbance associated with a human activity sort of fits in to the natural disturbance climate, if you will, in an ecosystem. Is whatever it is that we're doing similar enough to natural disturbance that the ecosystem can accommodate the additional input? That is often the big question. All right, who in the room remembers what is happening in the left-hand photograph? Yeah. Ripple Rock, good for you. I love it. It's hard to find people that still remember. Late 50s, the largest, how did it go? The largest non-nuclear explosion in the history of the Earth. They blew up a reef in the middle of Seymour Narrows up on the Inside Passage. One of the things that is important to recognize is not just that disturbances are important and not just that they cause interesting responses on the part of ecosystems, but that there are species whose very existence relies on disturbance. And these are three familiar examples from the terrestrial realm. Fireweed, self-explanatory, tends to come into burned areas right after the fire is out within the first year or so. The same story with avalanche lilies in areas where avalanches are common. Anybody who's been around this part of the world for very long knows that when there's a clear cut out in the woods, you add about 2 years to the end of the operation, and you've probably got a really good blackberry patch. Good enough to get enough berries for at least a couple of pies for a morning's effort. Again, a species that is exploiting disturbance and probably either would not exist at all or would exist at a much more minimal level without the disturbance. So the pitch is the disturbance is normal. The issue is to what degree does anthropogenic disturbance sort of mesh with the normal disturbance regime in the ecosystem, and to what extent does that differ? So with that, let me get a little more specific now and talk about our objectives. We recognize 5 types of disturbance that occur in the typical culture cycle in the Guido Aquaculture operations of Puget Sound. Predator exclusion structure placement, followed by the period of time during which those structures are present, removal of the structures, and then the time period following that when we have very high densities of relatively large geoducks within the cultured plot, contrasting with somewhat lower densities elsewhere, and then finally the harvest disturbance at the end of the story. Our emphasis in the early part of our project has been on harvest disturbance. That's really what we're focusing on. The main reason, of course, is that was the process identified by the state legislature as the #1 priority research topic. And it was from those good folks that we received our funding through the equally good folks at Washington Sea Grant. So we are interested by disposition, but also by virtue of the priorities of the funding organization that is supporting us. Okay, so we are focusing on benthic fauna, and I think it might be worth saying a little bit about what benthic fauna really are, because if you haven't thought much about this part of the biota of the world, it's a bit of a foreign area, and it's worth saying a bit about it. These are animals that live either on or in sediment, and in the case of Geoduck Aquaculture, we're talking mainly about sand and silty sand, that sort of range of sediment types. Most benthic fauna are invertebrates, although there are a few vertebrates that show up, and we'll talk about that a little bit later. The group as a whole tends to be quite diverse. It involves a number of different taxonomic categories at all different levels of organization and a very large number of species. It's important to realize that benthic sediments are the largest habitat type on the Earth that involve any sort of solid substance. Second place is way far back, so this is a pretty important type of ecosystem. The dominant groups in nearshore cases, such as the intertidal sand flats of the South Sound, are usually crustaceans, polychaete worms, and small bivalves, and that's pretty consistent across the board wherever you work. The densities of in this community can be extraordinary, often far in excess of 10,000 individuals per square meter, sometimes 100,000 individuals, and we have encountered densities approaching that more than once in our own project. The animals are typically small, which sometimes gives to the mistaken impression that they're unimportant ecologically. We beg to differ. And by definition, the group that we're working with is that group of organisms that is retained on a 0.5 millimeter screen when you pour your sample through the screen. In the end, it's all determined by size, and that's obviously quite arbitrary, but that's been the standard cutoff point for the type of fauna that we're working on for quite a long time. Okay, we have identified 3 sites chosen with the assistance of the growers, and we are extremely grateful for the ongoing cooperation that we've received from the growers. We'll name some names at the end, but I'll keep it at that for now. The priority was to identify sites that were going to be harvested fairly early on in the timeline of our project so that we could get some data before the harvest occurred, sample through the harvest event, and then have a substantial period following the harvest in order to sample and look for patterns of response, and then to have the time to process all the samples, work up the numbers, write the reports, and get things published so that everyone can have access to the information. And as things have worked out, these are the 3 sites that we've settled upon, known as Foss, Mankey, and Chelsea Wang. Foss and Mankey are on opposite sides of Case Inlet, and Chelsea Wang is down in the outer part of Eld Inlet. Now, we also have study sites that we're using for work on the structure effects, but we're not going to be talking about that tonight because we don't have any data to report on that specific part of the project as yet. We do have some information on the harvest part of our study. Okay, this is a graphic that shows the setup of our study areas, and it's in 2 parts. The upper part shows sort of the standard scenario where at each site we have a treatment plot and a control plot. The plots are, we've tried to make them at least 2,500 square meters in area. The exact shape varies somewhat depending on the configuration of the site. Some of them are sort of long and narrow, other ones are a little bit shorter and a little bit broader, but the area is pretty much the same in all cases. One of the plots is what we refer to in the graphic as the treatment plot. This is a plot in which cultured geoducks have been emplaced, and they're in the grow-out phase approaching harvest. A control plot is relatively nearby, and it needs to be relatively nearby so that it can be as similar as possible in terms of habitat characteristics, with the obvious exception of the fact that there aren't geoducks planted in the control, whereas there are in the treatment. So we have the 2 plots, a control, which is ostensibly an undisturbed nearby plot of similar habitat type. The treatment plot, which is the cultured plot, has a high density of outplanted geoducks that are approaching the termination of the culture cycle. In addition, we have 2 transects running outward away from the edges of the treatment plot in opposite directions. These are 50 meters in length, and you can see down here in the detail that each of them is sampled at 2, 5, 10, 20, and 50 meters distance from the edge of the plot each time that we go out and do a sampling routine at a site. Okay, and again, this of course is not to scale. The scale is pretty whacked out here. The sampling involves 2 categories of infaunal sampling. One is core samples, shown by the small circles. The core samples are distributed within each of the 2 plots based on a draw of random numbers so that the placement is unbiased. The core samples are small. They're about 5 centimeters in diameter, sampling an area of about 20 square centimeters. But we take 10 during each sampling event, in addition to the groups of cores that are taken on the transect. So we end up taking, I think the numbers add up to 50 cores per site per sampling event. In addition, we're taking larger samples that we refer to as excavations, shown here by squares, but they're actually circular as well. And basically, they're the diameter of the bottom of a 5-gallon bucket. We believe in only using really high-tech equipment in our study. So we have a bucket with the bottom cut out. It works great as a coring device. The purpose of that type of sampling is to try to get a better handle on larger animals that are less abundant and are not really appropriately sampled by the coring activity. So the larger excavations are screened through a mesh that I think is 5 millimeters. Is that right, Sean? 5 millimeters mesh size, and the core samples, where we're really focusing on the smaller organisms, The screen size is 0.5 millimeter, as I mentioned earlier. Okay. So that's the setup. So now what I want to do is give you a quick look at our inventory of samples, and this figure will take a little bit of translation, but hopefully it'll work out. So from now on, we're talking mainly about data from the FOSS side, because that is the one where we're farthest along. The reason is that FOSS was harvested in November and December, so we wanted to get as much information from FOSS as we could before the harvest occurred. So this is the timeline starting in May of 2008, and that's when we got our money right there. That was a big day. It was a big month. We began sampling in June, and then these are monthly intervals through February of 2009, which I think we're still in, just barely. The red circles below the dates indicate that sampling was done at some time during that month. I think actually the August sample actually was done on the 31st of July, so I had to cheat a little bit just because of the space available on the slide, but it was basically a month apart. So we've sampled every month from June until this month, except for September, and we skipped September because the tides were really crummy. We need good low tides to work. September and March don't give us very good low tides, so those are our, that's when we all go to Hawaii and drink, whatever. The green dots indicate samples that we have completely processed. And this, I hope, will be a little bit sobering to you in terms of the challenges of actually doing this kind of work. The samples from the cultured and control plots are finished through November, which is just about the time that the harvest began. The samples from the transects at FOSS, we've only been able to complete those collected last June and July. We've not sampled the other two sites nearly as intensively because the harvest is not scheduled until sometime in the coming months. So we're sort of ramping up our efforts now at these two sites, but to this point have not done a great deal of sampling. As you can see, 3 sampling events at Mankey, and we have only 1 set of cores from treatment and control plots that have actually been worked up, and none from the transects. And then just 2 samples taken so far at Chelsea Wang, and we've not had the opportunity to get any of those worked up. Okay, why is it taking so long? Well, it just does. It's as simple as that. This is a breakdown of the time required to work up a core. We collect 150 of these a month when we're in our normal sampling routine. It takes about an hour to screen the core and then rinse what's retained on the screen into a jar, and then pick through that material to separate the animals from the debris, from the broken shells and the sticks and all the other stuff that shows up on the screen. It takes about another hour to actually identify and count the animals. And then it takes something on the order of 15 minutes to a half an hour per core to complete our quality assurance, quality control procedures, and then a few extra minutes to enter the data into the appropriate database system. The total per core is roughly 2.5 hours, okay? And that works out to about 450 hours of work per month, which is enough to keep 2 or 3 people busy full time. And we do not have that level of human resources working for us, unfortunately. We're certainly not complaining about the support that we have, but clearly there's a lag. And so one of the things that we're doing right now is trying to develop strategies for selecting those samples that will be most informative in terms of the things that we're all interested in, so that we can get some results out to people sooner. But this is probably the fundamental challenge of the project. We don't really mind going out on low tides in the middle of the night in December when it's snowing. We can handle it. We did it. It doesn't last very long, and then you get to go home and go to bed and you feel okay afterwards. The real problem is just grinding through these cores. It takes a lot of time and effort, and it takes the right kind of person to do it, because it's a very monotonous and sort of brain-numbing process. It's not something that just anyone can do day after day. Now, before we go into looking at the data, I wanted to also give you a little bit of perspective on our funding situation. We originally envisioned this as a 6-year project, obviously assuming the blessing of the legislature and their funding as we go along. So what we've done here is list out the major topics that we're looking at from year one, which was actually, year one was just a partial year. It was the last part of fiscal year 2008. Did I get that right? 2008. May and June of 2008. And then we went into year two, which we're in right now. And then year three begins on the 1st of July, 2009. I hope you followed that. So our initial emphasis, as I mentioned, has been on harvest disturbance. We are going to be expanding our effort in looking at structure-related disturbances with regard to the infauna, with regard to epifauna, and also with regard to mobile organisms that are attracted to the structure fields to try to get better information on what is attracted, what might be repelled, and how they might be interacting with the natural ecosystem. And then we have what we're called alpha sites, and these are sites that we were hoping to follow from start to finish through an entire culture cycle, which of course takes 5 or 6 years and is only attainable with funding all the way through. Well, the reality is that we're in year two. Year one was a partial year. We have money for this year and we have money for year three. But after that, we don't know. And you all know what the fiscal climate is like right now and what the attitude is in Olympia with any piles of money that are sitting around. They get gobbled up really fast. So we're very concerned about this because if, in fact, we do get cut off at the end of year three, and I don't want to sound alarmist, but it's certainly a possibility, then that is going to greatly foreshorten our project, and it's going to really cut into what we're able to do. So I just wanted to make you aware of that there are dire consequences for this project that are emanating from the much larger financial crisis that is sweeping Olympia along with every place else. Okay, now some data. But first, some reminders. Okay, these are preliminary data. They have not yet been analyzed statistically. They've not been through all the phases of our quality assurance and quality control procedures. I hope you're catching my drift here. They've not been peer reviewed. The project team is not yet convinced that any of the apparent patterns in our data are in fact real. The reason we're displaying them is because we want you to see the kinds of data we're getting and to sort of open your eyes to the sorts of questions that we can address. The numbers are intriguing, but it's really important to keep all of those caveats in mind. So when you do see the data, don't get so excited, okay? Okay, so let's look at some numbers. So these data are densities for a group of species that we refer to as the Corophium group. It's actually 3 species. This one here, Americorophium salmonis, is the one that most of you have probably heard about because it's very important food for out-migrating salmon in Puget Sound. There are 2 other species that get into the mix. These are both introduced species, actually, but they're fairly widespread and commonly encountered in our samples. So we're counting them as a group for now. These are tube-dwelling amphipod crustaceans that are about 1 centimeter in length when they're full grown. So here's the story. So in the plot, the sample date is shown across the horizontal, and the mean number of individuals in our cores extrapolated so that the number is an estimate of the density per meter square is shown on the vertical. The blue bars represent data from the cultured plots. The red bars represent data from the controls, and this is just for FOSS, 1 site. Okay, 5 samples at FOSS, 1 site. The last set of samples included some cores that were in a part of the cultured plot that had been harvested, I think, just the day before we collected the cores. So there may be a signature of harvest in here, there may not be. But the pattern that is apparent in this particular set of data is that densities of Corophium in the control area tend to be higher than those in the area that it is being cultured. Another abundant species. This may be the most abundant species in the system at FOSS. This little thing, Microphthalmus hystrix, polychaete worm, with a maximum length as a full-grown adult of about 2.5 millimeters, about 1/10 of an inch. And there are hundreds of them. And just because they're small doesn't mean they're not important. Keep that in mind. You can see that the densities have reached as high as nearly 40,000 individuals per square meter. The pattern is very much the same for Microphthalmus as it is for Carophium. It appears that they do much better in the control site than they do in the cultured site, subject to all the caveats that I just listed out. Okay, we've also looked at some grouped taxa. Now this is a plot that shows all of the infauna lumped together in just one big pile. Okay, so it's the total number of all species put together. And you can see that the densities range up to about 90,000 per square meter. The same basic pattern seems to be apparent. The numbers are a bit higher in general in the control plot than in the cultured plot. The story is a little bit with all crustaceans put together. Okay, so now we've excluded the worms and the little clams and everything else, and we're just talking about things like amphipods, small shrimp, and that sort of thing. There's not really a clear pattern here. Here's a sample where there seemed to be more numbers in the cultured plot, but in the others, the numbers are somewhat higher in the control plot. So a clear pattern is not apparent in that representation, and that clearly is something that we'll have to wait for a good statistical analysis. The polychaetes, again, are showing that pattern that we've seen already, and that's probably what is driving the pattern in this plot, which includes the polychaetes. They probably are the dominant factor there. So it's a little bit redundant. Bivalves, these are small clams that we find consistently in the cores. Far less abundant. You'll notice that the densities are quite a bit lower. And here we're talking about species that typically most of us are not familiar with, things like Rochafordia, Talina, Micella, things like that are quite small. We've not seen a single juvenile geoduck in any of our cores, to our great frustration. We've been looking, but we haven't seen a single one. The story here is not clear. That one site or one date where there seemed to be a lot more in the culture plot, but another date where it appears to be exactly the opposite. Okay, we've also set up some very simple indices of biodiversity, recognizing that the impact of geoduck aquaculture on the biodiversity of the resident ecosystem is something that everyone is interested in. And we've used two of the simplest indices. There are a number of different ways of indexing biodiversity, and it's actually a fairly active area of research and argument. It seems as though the diversity index that people choose to use is often that is most consistent with their particular perception of the system. So it can be a bit of a problem. But I've just used two simple ones here because we didn't have a lot of time to crunch the numbers before this presentation. This is the simplest of all. It's simply the total number of species in each sample. Okay, and here we see that on most sampling dates, the diversity according to this index is very similar between cultured plot and control. Now, there's something in this presentation for everyone. I know those of you that are into geoduck aquaculture will be happy to finally see something where the blue bars are taller than the red bars, and that is the second diversity index, which we refer to as evenness. This refers to how even the distribution of individuals is among the different species. So if you have 10 species and each one you have 10 individuals, that's a very even sample, and that is generally viewed in the ecological literature as being a system of high diversity. If you had the same number of individuals but only 3 species, and the distribution was not even, then the evenness would be substantially less, and you would view that as somewhat less diverse. So at least with regard to this specific index, we see that as the summer moved into fall, it appears that the evenness in the cultured plots was somewhat greater than that in the controls. Okay, one slide to look at the Transect data. Again, this is data just from the FOSS site, and we only have it worked up for 2 dates, 20th of June and the 1st of July, so it's only a couple of weeks apart. In the first plot, and in both plots, the north transect is represented by the darker bars, the south transect by the lighter bars. We sample the same distances away from the edge of the plot in both directions. It's interesting in how much agreement there is among the density data at the different sites in the June sample. In the July sample, there's less agreement, but to my eye, no obvious pattern. There's some some locations where there's more adjacent to the control, some dates where there's more adjacent to the cultured plot, so no strong pattern there. So we don't appear to have much of a story yet in terms of the extent to which disturbance extends beyond the actual culture plot itself. That remains to be seen. Okay, let me give you just a little bit of insight into our quality assurance, quality control. We have a couple of hotshot taxonomists on our team. We like Jeff Cordell. As you can see here, you don't mess with Jeff. Okay, Jeff is a very good crustacean taxonomist. He doesn't suffer fools lightly in this particular area of endeavor, despite the photograph, which was pouring down rain and cold and miserable. He's a great guy. And he has been our mentor and our QA/QC guy for crustaceans. And he recently went through a number of crustacean samples for us. And these are a representation of basically everything that he did. So in the Corophium group, for example, he looked at 139 individuals and he found that we were identifying them correctly 97% of the time. For a Cumasian, Cumella vulgaris, we hit the jackpot. We didn't miss a single one. Now, turns out this is not actually Cumella. This is one of those last-minute dashes through the photos on the web. This is actually another type of Cumasian called lamprops, but it's very similar in appearance to Cumella. Gives you an idea of what it looks like, a couple of millimeters in length. Huntomania, which is a type of harpacticoid copepod, one of the most common species in our samples. He looked at 284 individuals, and we had it right 95% of the time. 93% accuracy with 291 other harpacticoid copepods, and this is what a harpacticoid looks like. Little tiny sort of sand flea type thing that lives in the sediment. Overall, Jeff looked at 784 individuals, and 95% of them were correctly identified. So we generally feel pretty good about that. There's always room for improvement, but it's not too bad. We're just getting started on QA/QC with our polychaetes. They can be quite challenging. I am worried that the numbers might be a bit lower, but hope springs eternal, so keep in touch on that one. Okay, finally, a few data from some of the larger species that are assessed using the excavation samples. I mentioned that occasionally one finds vertebrates in the Benthic Fauna, and as you probably know, sand lance, a very common fish in our region, although not as common as many of us would like, in fact, buries themselves in the sand, apparently when they're at rest, apparently to avoid predation when they're resting, although we don't really know for sure why they do it. But remarkably, when you're taking core samples, once in a while you'll get sand lance in your cores. You would think that they would just swim away. I mean, it just makes me crazy, but they don't. So the numbers are relatively low, but we do get them fairly consistently. So these are the mean densities per meter square. So we're talking about, a few individuals to a few tens of individuals. And it's interesting to note that they're pretty consistent between the control and treatment areas, except for this first sample. We started doing the excavation samples in October. The harvest event was in November and December. And we see that post-harvest, they hung in there pretty well. So it may be that based on this very limited window that the sand lance are relatively tolerant of harvest activities. But we'll follow that as we go along. A burrowing sea cucumber, interesting concept that is fairly common, sort of 10 to 20 individuals per square meter. And again, relatively abundant in both kinds of plot, not any particular sway one way or the other. Sand dollars, probably one of the strongest patterns in our data, and this is one that I think we probably would be willing to go a little farther with because you can see sand dollars. And the differences are pretty obvious. There clearly are more sand dollars at Foss, at least, in the control site than in the cultured site. And the pattern holds up with size data as well. This is the mean length of a sand dollar, which I'm not sure that we should call it that. I sort of like circumference. Anyway, regardless of what you call it, you can see that the sand dollars that we find in the control sites are generally substantially larger than those in the cultured site. Okay, so I'm going to depart from our project now and try my very best to pretend like I'm a geochemist. And this is a big challenge. I've been in this sort of situation in the past, and it's not always pretty, but I'll do the best I can. Neither Roger nor Jeff were able to come to the meeting, so they sent us a bunch of information, and here we go. So these guys are at the Center for Environmental Science at the Horn Point Environmental Lab at the University of Maryland. Great group of folks that have been out several times to work with us. So their goals are to look at the dynamics of nutrients that occur in what we call the pore water, the water that occurs within the sediment, that occurs between the grains of sand and the sediment. They're interested in the production and storage of inorganic nutrients in that type of water and the degree to which it exchanges with overlying water as the tide comes in and out. And they're particularly interested in how geoduck culturing activity might influence the dynamics of the exchange of these all-important chemical constituents of water between the sediment and the water column. Okay, you can probably read that just about as well as I can. So the idea here is to compare characteristics of the pore water chemistry before and after harvest. And then I also actually to try to sample right during the harvest to actually see what sorts of chemical constituents are sort of flowing down the beach as the water from the water pumps is being used to excavate the geoducks. Okay, so here's some photographs of, this is Roger. Some of you may know Roger Newell, is a very well-known invertebrate ecologist. And there are several devices here used to assess pore water chemistry and also to look at the rate at which the water that's in the sediment actually comes out of the sediment during low tide and intermingles with runs down the beach and intermingles with the moving water. Okay, so here's some data. And as Jeff has put it, they have two surprising results. One is that during low tide, most of the water stays in the sediment. Now, there's some exceptions to that, but their conclusion, based on what they refer to as PVC wells, basically they're just tubes that are placed into the sediment, and they're able to measure the rate at which water comes up into the tubes and then flows outward and onto the sediment surface, that in general, that flow is minimal. So the water that is in the interstices between the sand grades tends to stay there during low tide. Okay? The second part of this is that based on interpretation of pore water concentrations of iron, one of their conclusions is that there is very likely substantial nutrient regeneration occurring within the sediment, okay? And that they detected no sulfide. Sulfide's the stuff that smells like rotten eggs that we've all encountered in certain types of sediment, but they saw none of that at the Foss site. So the bottom line here is that the water is staying in the sediment at low tide. It's not intermixing to any great degree with the overlying water, and that there is substantial regeneration of inorganic nutrients happening within that pore water. So sort of the follow-on from that is the thought that, or is the question of how that dynamic will be changed as a consequence of actual harvesting activity. And hopefully they can nail that down this summer. Okay, so I'm going to stop now, finally, and Micah Horowitz is going to come up here.
Okay, I would like to shift your consideration from invertebrate animals in the sediment to a plant. So today I'm going to talk about local effects of geoduck aquaculture on a meadow of intertidal eelgrass in Samish Bay, Washington. And because Samish Bay is a long way from here, I'll introduce the site first. So Samish Bay is in North Puget Sound, actually just north of Puget Sound, about halfway between Seattle and Vancouver. And there are two things about it that are pretty different from all the sites that you've just heard about, and that make this study different because of it. One is that eelgrass occurs at this site, overlapping with Geoduck Aquaculture, and the other is that harvest took place on a faster time scale. So harvest took place in May of 2008, so we actually have amassed some post-harvest data, and I'll be showing those results. So this is a picture of the bay. It's a very extensive eelgrass meadow. When it goes down at low tide, it's just miles and miles of eelgrass all the way out. And just to talk about what that plant is, it is native to this area, a long-lived saltwater flowering plant. So it's not an algae. It produces pollen and seeds and reproduces both asexually and sexually. Asexually by spreading out like a grass under the sediment and then putting blades up, and sexually through setting out pollen and then seeds which can float and disperse. It lives both intertidally and subtidally, and all that I'll be looking at is intertidally because it's easier to access places where you can get just when the tide is down. It forms these meadow habitats, which I just showed the picture of, that are important for both infaunal animals and epifaunal animals, and may also be very important foraging grounds for fish of commercial interest. And finally, one of the reasons that it makes a distinct habitat is by reducing erosion with the roots and rhizomes of the plant holding the mud together, and also reducing wave action. So with the above ground parts of the plant, the leaves are ameliorating wave action and sheltering animals within it. Okay, so just to give a history of the site, this whole study was done in cooperation with Taylor Shellfish. They have one geoduck farm up there in a bar in the very middle of the bay. And so here, this is the area that was and is being farmed for geoduck. And as I understand it, when this area was planted for geoduck in the summer of 2002, there was no eelgrass there. So this plot had been leased and then planted with oysters for some time, but it wasn't working for them because in the winter they got washed off. And then it was decided to plant geoduck there. So that was 6 years ago, 6.5. And then the geoduck were planted in, as you've already seen from Glenn's presentation, in the predator exclusion devices, the tubes. And eelgrass then settled on the bar. So where it didn't used to be, it came in. But this cartoon is just supposed to illustrate that it came in patchily. So it didn't form the same lush meadow that you saw in the last photograph, but rather patches of eelgrass within bare mudflats. And this is over an area with geoduck in it. And also just to point out, with the design of the study, it's pretty similar to Glenn's in a lot of ways. This would be the treatment area. So in 2002, it was planted with geoducks. The tubes went in, then were taken out a year and a half later. And then the geoduck were allowed to grow until the summer of 2008 when they were harvested. And this is the control zone. So this is not farmed now, and it has not been. And we're sampling equally both inside and outside of the farm. And the placement of our samples is random within space. And I have reasons for doing that, which I'll talk about soon. You will notice that the control zone is only on one side of the plot, and that's mostly just for reasons of the topology of the site. There's a big channel here that is not intertidal. It always has water in it, and that's where you access the bar from the boat. Okay, so here's a picture of the geoduck harvest that happened in May and June of last year. Hopefully from this photograph you can see that this is the farmed area, and it is patchy. So this is on the first day of harvest before any harvest has really started in earnest. And there's eelgrass in some places, no eelgrass in others. And all the geoduck have been marked, and then they're being pulled out with spot stinger harvest. So where they've been flagged, harvesters will go in and then liquefy the sediment and pull them out. Tube installation happened later in the same summer. So the whole of the bar, again, had the predator exclusion devices planted in, and juvenile geoducks planted in after them. And then finally, the last aquaculture activity in this year was the nets being placed over the tubes, again to protect the juvenile geoducks from predators. And in between each of these distinct events, which Glenn usefully characterized as different disturbances, we had sampling dates. So before harvest, immediately after predator exclusion devices were installed, and then after the nets were installed over everything. And so that's 4 dates. The first thing that we wanted to monitor, because the bar started out with patchy distribution of eelgrass, was whether that has changed. And so all of the data that I'll show tonight are going to be roughly in this format with 4 sampled dates. At each date, there's the not farmed area being compared to the farmed area, and there have been statistical analyses performed. So you'll see those too. So you can see that before harvest, the patchiness was pretty evident. Just with our random placement of plots, about half of them within the farm zone landed on eelgrass, and all of them outside of the farm zone landed on eelgrass. There's no patchiness outside of the farm. It's a solid meadow. Immediately after harvest, there was a slight increase in the number of eelgrass patches that our samples landed on, roughly the same after the installation of tubes, and a lower proportion after nets were installed. And none of these were statistically different. Okay, but then looking at the places where eelgrass did occur, so setting aside the fact that it's patchy within the farm and not patchy outside, we wanted to look at the density of the eelgrass shoots in both the treatment and control areas. And this is just the way that it's measured. In a 1/2 meter by 1/2 meter quadrat, we counted every blade of the grass in all of the 25 plots inside and outside. So this, before it becomes too dazzling, I just want to point out that this is why we took a lot of pains to be spatially explicit in our sampling, to look at whether there are any spillover effects from geoduck aquaculture outside of the harvest and culture zones. So for every random sample that we collected, we knew exactly where it was placed relative to the farm, and then we're able to look at whether there are different intensities of effect or changes that you can notice in the unfarmed zone from each of these events, the harvest, the tubes, and the nets being installed. And so here on this x-axis is just the distance from the harvest boundary. All the blue points are inside the farm zone, and all the green points are outside the farm zone. And what you're looking at is eelgrass density. So at none of these dates is there an evident pattern. Before harvest, it didn't really matter how close we sampled to the farm zone. The eelgrass density outside of the farm zone was the same, and this was the same for every date. You will probably notice that there's a lot of seasonal change in density, and this is typical for eelgrass in Samish Bay. With the grass being larger as plants and more dense in the summer, and smaller and sparser in the winter. So you can see that both the farmed and unfarmed zones had decreases in density at our winter sampling date. But again, looking at something besides just the spatial distribution, considering the average density within the farmed and unfarmed plots in aggregate, we look at it this way. So again, the green bars are data from the not farmed area, and the blue bars are from the farmed area. And before harvest, there was no significant difference between these two areas in density. So wherever eelgrass did occur within the farm zone, it was of equivalent density on average as outside the farm zone. And after harvest, a difference emerged with eelgrass of being lower in density within the farm zone. And this difference persisted across all of our post-harvest sample dates. And you can see, again, that both the not farmed and farm zones show this seasonal pattern of increase at the height of summer and then decrease in density towards the end, or towards winter. Okay, and then we were also monitoring the morphology or size and shape of the grass. And I'm just going to show one measure of the grass. This is the sheath length. So this is from the internode, the first internode of the plant, to the place where the leaves start to split apart. And it's a pretty good proxy for just general plant size. So we measured this for thousands of plants. And you can see, again, this general trend of larger plants in the summer, smaller plants in the spring and winter. And this is the one case in which there was a difference between the farmed and not farmed plots before we began. So the shoots were slightly larger in the not farm zone as compared to the farm zone to begin with. And this difference stayed true at every date afterwards. Okay, I mentioned that eelgrass can reproduce asexually and sexually, and we wanted to monitor this behavior of the grass to know if there were to be any reduction in coverage or density, how the eelgrass would be able to recover. And one thing to monitor is to look at the capacity of the plant to undergo sexual reproduction. So this is a picture of the spathe of the plant, and in there are flowers that may produce seeds someday. So here, there's a season of flowering, as there are many things in eelgrass, and there are a very few flowering shoots evident in the first sampling date before harvest, but the differences in the occurrence of those flowering shoots was not different between the farmed and not farmed zone. This is the rate of flowering, so it's standardized to the density of the plant. So this doesn't reflect any of the differences that we found in density. At the second immediate post-harvest date, the plants had really ramped up production of flowering shoots, and there were, again, no differences between the two, control and treatment. And then in the late summer, a difference emerged between the farmed and not farmed plots, with the farmed plot post-harvest showing a significant decrease in the rate of flowering among shoots. Okay, this again is a geoduck pothole that you saw other pictures of post-harvest. And we are looking at a couple of abiotic or physical characteristics on the bar. And one is sediment organic content. So this can be an important aspect of habitat for all of the in-fauna, which we are also measuring, but those data are not complete yet, so they won't be presented tonight. So for sediment organic content, before harvest, there was no difference between the two sites. After harvest, the not farmed area had significantly more organic content than did the farmed area. And this was true for all three post-harvest sampling dates. We were also looking at change in elevation across the harvest zone, just because we were interested in whether harvest or the presence of eelgrass might affect erosion. And so just to describe how this is done, these are rebar stakes at set intervals that I've installed. And if you run mason line across them very tightly, then you can go at set places and measure the distance from the mason line to the ground. And the rebar stakes are very stable, so you can know that you're comparing the elevation of the same place at different dates. And we ran a transect of these rebar stakes across the harvest boundary so that we were again looking for spatial patterns in a change in elevation to see whether there might be larger changes in elevation nearer the harvest zone. So here is that first answer to that question. Here are points that are representing the change in elevation since harvest in the not farmed area. And here are points representing the change in elevation since harvest in the farmed area. This red line represents 0 or no change. So you can see that there's lots of variation. Some places have gained slight amounts of elevation. This is in centimeters. And some places have lost it. And you can also see that there's more variation within the farm zone than there is in the not farm zone. And I think this just reflects that the geoduck potholes are a very spatially specific disturbance. So depending on whether or not you're sampling right over one or away from one, you can either get very small changes in elevation, or I sampled here, so I know that this represents a couple of different potholes where you're actually seeing these big dips in the sediment. And this is 60 days after harvest. Yes, but there's no evidence at all for a difference in the change of elevation as you near the harvest zone. So for changes in elevation, there's no evidence of a spillover effect. Here is looking at the data in aggregate. So since you were just looking at the raw data, you know that this is masking a lot of variation. But at both 30 and 60 days after harvest, both the not farmed and farmed areas demonstrated significant reductions in density or in elevation on average. So this is a loss on average in the not farm zone of about half a centimeter and of about a little more than 1 centimeter in the farm zone. And that difference is statistically significant. At the 60-day after harvest sampling date, there was no significant difference between the change in elevation in the farmed and not farmed zones, but both remained significantly lower than pre-harvest. Okay, so just to sum up farming effects to date, I'm going to show kind of a schematic diagram of the settlement of eelgrass onto the bar, changes since harvest, and all of our findings, just summed up in sentences. So here is just eelgrass density. There are no units on this graph in the farm zone. Before geoducks were there, I understand there's no eelgrass. The first crop was planted in the summer of 2002. Eelgrass settled on the bar and came to some patchy distribution and density right before harvest, which is what we sampled. Then harvest occurred. We sampled afterwards and noted a significant reduction in eelgrass density. This reduction continued in our third and fourth sampling dates after the installation of tubes and nets. And so that describes the general pattern in eelgrass density. The things that we found immediately after harvest were the reduced density, reduced organic content in the farm zone, greater elevation loss in the harvest zone than outside. At the second date, reduced density, reduced organic content, elevation loss, and reduced rates of flowering. And at the third date, reduced density and reduced organic content. So the other results that we have are that there was no change in patchiness from one date to another. So there's not a difference in the number of quadrats that we're laying out and finding eelgrass inside the farm zone. And there's no evidence of spillover effects, no matter how close you get to the farm zone. And so as this work continues, we're going to get data on the infauna at this site, and we're also going to be looking at patterns of change in the eelgrass going forward for the next several years to see how it settles back into this area, which now has tubes installed. So I'd like to thank Sea Grant for funding and the cooperation of Taylor Shellfish, especially for transport out to the bar, and a lot of field assistance and useful advice that I received. And now I'll give the podium back to Glenn.
So I just wanted to wrap up, and many of you are probably aware that Sea Grant has funded several projects, including Micah's project and ours, and also the project that's being done by Roger Newell and Jeff Cornwell, in addition to some other research work. And I just wanted to take the opportunity on behalf of everyone that has benefited from this funding, and and to mention that in particular Penny Dalton, Rachel Waters, and Teri King, all the help that they've provided to us, and to Teri in particular for organizing this event. So sincere thanks from all of us to the Sea Grant staff. On behalf of our project, the project that I spoke about earlier, we wanted to thank all of the collaborating growers and tribal biologists, homeowners, and other colleagues that have helped us get going. I'm sure that we've left someone out, I have no doubt, but I know that some of the folks in this list are in the room, and I want you to know how much we appreciate everything you've done. I have a feeling that there have been times when you have viewed us as royal pains in the you-know-what, in terms of all the stupid questions and all the following around and everything, but we're extremely grateful for all of your help. So with that, I think we're finished, and I would like to open it up for questions and discussion. I'm good for as long as you folks are. You know, we may need to finish up in the bar, but one way or another, I'm more than happy to address questions. I did want to mention that Sean McDonald is here in the front row. Sean, wave your hand. Sean is a postdoctoral researcher working with us on the project and has been a crucially important part of our program from day one. And he gets tired of me saying that we couldn't have done it without him. But I think he likes it, you know, way down deep. So anyway, with that, thanks very much for your attention, and we're happy to answer questions.
You had mentioned at the Foss site that there was quite a difference in the abundance of sand dollars. between the sites, and I was wondering if you knew, was that site cleaned before the tubes were planted?
I actually don't know myself. It was not. Okay. So that's a difference that existed prior to the culture activity. Okay.
Thank you.
Yeah. No, we'd actually talked about that the other day, and I wasn't sure what the history of that was. So.
I just had a general question. Could you tell me how long something like Corophium or the polychaetes live, how quickly they turn over?
Oh boy, good question. Anybody have a banjo? Yeah. A lot of the amphipods live on the order of a couple of years. I honestly don't know the lifespan for Corophium, but it gets to be fairly large as amphipods go, so I would suspect that it lives for at least a year or two. Sean, do you? Yeah, you're not sure. But that would be fairly typical for amphipods, is sort of a year or two. The polychaetes, as far as I know, are all over the place, and I strongly suspect that little thing, that microphthalmus that we see so much of, is probably relatively short-lived because it's so small. Often, although not always, the smaller the animal, the shorter its lifespan. And I would be very surprised if it was not the case for that species, but we know very little about that species despite its extraordinary abundance. And I should mention, just as a follow-on to that a lot of the species that we find don't even have names. Some of them haven't even been described. So, we're sort of working against that sort of information gap for a lot of the species that we're working on. This one up front.
Thank you. Is this thing working?
It is.
Okay. Realizing, of course, that your real focus is on benthic critters, your South Puget Sound sites, all three look like they could have an association or be identical to spawning sites for surf smelt and sand lance. Mm-hmm. Have you tried to take that into account in your study?
Well, we've talked about it. We haven't developed any sampling specific to that issue other than the excavation data that we mentioned, and in those cases that the appearance of sand lance in the samples was sort of a surprise, but we certainly weren't targeting sand lance. My understanding is that our study sites are in general lower in the intertidal than the preferred spawning habitat, at least for sand lance. I don't know for surf smelt. Do I have that right, Sean? So we're probably not in a very good position to assess that question. It's obviously an interesting question and one that a lot of people worry about, but I think the position of our study sites is such that our data probably won't be of much help in that regard.
Are your sites full cover sites or just merely casts? Are your sites?
Well, the three sites that I talked about tonight, of course, are well beyond that. They're down at the end of the culture cycle. I haven't been out of Rogers since they put the tubes in.
They're all full cover.
Full cover? Okay.
Okay, thank you.
Yeah. Sorry, I can't give you a better answer to that, but that's an issue that we're not in a very good position to deal with.
Yeah, I would mention, just to follow on to that, our protocol for processing our core samples is compatible with other protocols for actually detecting the eggs of forage fish like surf smelt and sand lance. Although we're probably not in the preferred spawning area, we could detect eggs if they were present.
Have we seen any eggs? We haven't seen any eggs. Okay. Kirsten.
So you mentioned a few invasive species. Now I know it's hard to know, especially with these little guys, how many of them are native or not. But I was curious, in the biodiversity metrics that you looked at, was that all species, including potential?
It was the whole
native
works.
and non-native. Okay.
Yeah, we didn't make the effort to separate them out. Yeah. And you're absolutely right. We really don't know at this point how many non-natives we have. Obviously a very interesting question.
The question was for Mike on the Samish Bay site. Was the absence of eelgrass consistent for the non-farmed site and the farm site prior to the planting? So there was no eelgrass out there in that entire area.
No, that is not. I was not there, but that's not what I've heard. Rather that it was an extensive meadow of eelgrass, like in the picture, but that it was Fisk Bar that had no eelgrass prior to the installation of tubes. So Fisk Bar is the sandy portion right next to the channel, and that's where there was none, and then it became patchily settled in.
It was in the area that you were identifying as the non-farm site. It was already there.
It has been there for some time.
Next. Oh, Gordon. Hey.
That's for Mike here again. It appeared at the Fisk Bar in Samish Bay that your control was further from the channel, and so it would be actually interesting to know what happens in the channel, whether that patchiness. I mean, maybe it's a decline of patchiness as you go into the channel, because you've got a significant physical difference on one side and goes through i.e. the channel, then it goes through the farm and then further away from the channel. Mm-hmm. So I just wondered, maybe that was just a natural climb.
I know from walking around there, I have not collected data on it, that within the channel there is eelgrass that's quite dense, that is never exposed to the air because the channel is permanently sheltering it. So I guess Fisk Bar is surrounded on two sides by eelgrass, but on only one side is that eelgrass intertidal. So I've not seen, but also I've not quantified whether the eelgrass in the channel was patchy. The only eelgrass I've seen that is patchy is on Fisk Bar.
Anyone else? Oh, John.
It's just sort of a speculation. I've always was taught to look at a sandy beach as sort of a river of sand that was slowly flowing within the drift cell. And so I've sort of been wondering if geoducks, because of their size, actually, as they mature, would tend to stabilize the sand that they're living in and actually make it perhaps a more stable environment than your control, where the sand is moving all the time. It's just a speculation.
Yeah, and certainly reasonable. We do have, Micah made reference to the fact that if you put in a good rebar stake, it's a very stable reference point for changes in elevation, and we've marked our sites in a similar way in general. We have not measured changes in elevation, but I'm not aware of anything obvious in the way of changes over the time since we've started. Now, maybe over a longer time period, there could have been. Yeah, I mean, if some of the differences that we mentioned turned out to be real, based on statistical analysis and all that other stuff that we want to do before we put any stock in it, that's certainly an issue that we need to consider if there are differences between the two plot types, is the extent to which the ducks are stabilizing the sediment. I think it's a perfectly reasonable mechanism that could be used to explain some differences.
More eelgrass questions. In setting up your plots there, was any attempt made to allow for prevailing winds and current patterns?
No.
And if that was grid north on the image you showed us, is the prevailing wind there west-southwest?
I don't know what the prevailing wind is. The selection of the control zone was just based on the availability of a large swath of eelgrass right next to the farm. And they are of almost exactly equivalent tidal elevation. But in terms of prevailing winds, I don't know which aspect of the site is experiencing different pressures.
Also, if there's going to be a difference in elevation, will that really be perceptible until after the storm cycles in winter?
It may not be. So looking at these differences in elevation statistically, there were significant reductions in average density in both the control and harvest treatments. But they were very small and very variable. So some areas lost a lot of elevation and others not very much, but we're continuing to sample changes in elevation throughout the seasons. And Samish Bay does experience more intense storms in the winter than in the summer.
Now, one more question, we'll leave it alone. One hotly debated issue in the SARC panel was on the size of buffer zones. And it appeared like the scow might be parked on top of your control patch there, because you never park your boat on top of your product, right? Mm-hmm. So were any of your sample areas within the work area where they're physically parking the boats, stockpiling equipment, paths of entrance in and out of the work area?
That's a very good question. The scow was there was never any disturbance while I was there of the control area. No one ever walked on it or did any activity. But aside from the installation of tubes and nets and actual harvest, another disturbance, which I don't think any of us talked about, is just the human activity of walking around on the bar and dropping equipment. So as tubes are installed, you have giant bags of tubes behind you and are just moving along the bar. And the scow at some times was parked on portions of the farm. But as the farm got filled up again with the reinstallation of tubes and the nets, there was no longer any equipment left on the bar. So it was just kept off in the water so as not to interfere with any of the aquaculture activities.
Any last questions? No. Well, I'm going to give this to Penny Dalton, and I'm going to let her finish up the night.
I just wanted to add my thanks to a couple of the people that we haven't recognized yet tonight. One was the state legislature that actually sat down and hammered out a really amazingly good research bill that we actually were able to take to an RFP process and get good proposals from it. Not a very common thing to have a bunch of legislators write research priorities, but they did a great job, led by now retired Representative Pat Lance. The other thing, we worked closely with the Shellfish Aquaculture Regulatory Committee to develop the program. And I know that they had a lot of internal disagreements, but they were absolutely thoughtful and worked very well with us in terms of giving us suggestions for how the program would set up. The Department of Ecology, Perry Lund, has been our hero in terms of working through things. Gene Koenigs and Tom Klingman and a host of other state agency people. This has been a really unusual project for us, people that are used to working with science and scientists, because there have been so many other people involved constructively in it. So I just didn't want to let the opportunity go by without recognizing them. Thank you.
So I think we're done, and I just think we should give Glenn and Micah another round of applause. I want to thank all of you for coming tonight. If you're interested in participating in future sound science seminars and you're not already on our mailing list, there's blue cards outside. Just pop your email address, and if you have any ideas for future topics, our next one will probably be in April. I think we have a topic, but I'm always swayable. We've got some great research going on at the University of Washington, and it's always a pleasure to be able to bring the researchers down and share what they're doing in our community with us. And it's always a good night to come out and enjoy things. So thank you all for being here, and have a good night.